So this session is going to be broken into two pieces. One will be clinical. Will be my piece, and then the second piece that Joel will do will be some of the more basic science. And there will be a Q&A afterwards. So overview for my talk, we'll discuss very briefly what a prion is, because Joel's going to go into that in a lot more detail. We'll review some of the demographic features of prion disease and review some of the diagnostic process that we use clinically to diagnose the illness. And then we'll talk a little bit about why there's so much clinical variability. If you're in the sporadic workshop, you heard very different stories about different family members and what they went through with their loved ones. So first off, of course, we're all here because of this thing called a prion. A prion stands for something called a proteinaceous and infectious particle. It is essentially a transmissible protein, and it is the cause of prion disease. There's nothing else that is involved that causes it. We all make normal prion proteins. That's the structure on your left called PRPC, which stands for cellular prion protein. You see it has a lot of these kind of helices, and that's kind of the foundation of the normal prion protein. We don't know exactly what it does, but it has a couple of functions, such as neuronal housekeeping functions. It binds to other molecules, but it's supposed to be there, and it's supposed to serve useful purposes. But in prion disease, unfortunately, its 3D structure changes shape. It causes more of those kind of blue plated sheets, is what we call it, and that gives characteristics to the disease-associated form of the prion protein, which in our field, and what you'll hear multiple times this weekend, referred to as PRPSC, which stands for the scrapie protein, because of the prion disease in sheep and goats called scrapie. And underlying all of this is something called the prion paradigm, which basically is we all make the normal prion protein, those yellow dots. When they come into contact with the abnormal form, those abnormal forms convert the normal prion protein into itself. It's kind of like the bad kid in the schoolyard, where they kind of recruit more and more bad kids into the gang. Unfortunately, that's what happens here. And each time the normal protein gets converted into the abnormal form, it makes these kind of rods or fibrils that then can break apart and cause more seeds. And if this keeps happening, you'll get more and more of the abnormal prion protein being recruited. Of course, not you all, but in the general population, most people think of prion disease and they think of the acquired forms, right? And you can see in this chart that's well under 1% of all cases, thankfully. 85% of all prion diseases are due to what we call a sporadic etiology, meaning it just happens. And there's two possible explanations for that, and they're not mutually exclusive. One is it's just a spontaneous change of that normal protein that we all make that causes disease. Or it could be that instead of inheriting a mutation from your mom or dad, that later on in life, that cell in your brain develops a mutation and that causes the abnormal form of the protein. Both could occur, although it's probably most of these diseases are due to just a spontaneous change in the prion protein. And in our field, unfortunately, we like to name things a little funky. So we have sporadic CJD, variable protease-sensitive prionopathy, sporadic fatal insomnia. They're named differently just because they look different clinically and also underneath the microscope, but they're all due to the same thing, and that is a sporadic form of prion disease. About 10% in our country are due to a genetic mutation of the prion protein gene. Most of these affect about half the family. Autosomal dominant, so you only need one copy of the mutation to cause the illness. And of course, they also have different names like genetic CJD, Gershman-Straussler-Schenker disease, and fatal familial insomnia. I'm not going to talk too much about the genetic forms of the disease because you're going to get a healthy dose of that tomorrow morning with Simon Mead's talk, and he's going to present that in a very understandable way. Of course, the ones that we hear the most about in the media are thankfully the least common. That's the acquired forms, including Kuru, iatrogenic CJD, which is another way of saying medically transmitted CJD, or variant CJD, which is what we call a CJD in an individual due to eating meat contaminated with bovine spongiform encephalopathy, otherwise called mad cow disease. This is a curve that you might see frequently throughout the day tomorrow. It's called a Kaplan-Meier curve or a survival curve. On the X-axis is years or time, and the Y-axis is basically likelihood of survival. So if you start at the top, that 1 means 100% of people are living, and then as you go across the X-axis, that's the different percentage of people that are still living. So this is basically just an example of people with sporadic CJD. Mean illness duration from symptom onset until someone passes about 4 to 6 months. Average time from diagnosis to passing is about 1 month. As you can see, though, there's a lot of variability. That curve goes out pretty far, although if you look at the 1-year mark, about 80%, 75, 80% of people have passed within 1 year. But we do certainly see people that live longer than that. So a lot of variability, which will be a theme that we'll hear a lot about this weekend. Most people hear the 1 in a million number. That's what we call the incidence of CJD. It's really a public health statistic. It's very helpful for the epidemiologists and for us and for public health departments, but maybe not so helpful for families to understand how frequent an illness is. And I would argue even really not that helpful for a physician or a clinician to understand how frequently it is. So basically, it's the number of new cases of CJD that happen every year across the entire population. If you average that, it's about 1 to 2 per million per year, probably closer to 2 per million per year. But if you separate it by age, you see that that differs substantially. The number 1 risk factor for sporadic prion disease is actually age. So if you look at those over the age of 65, the incidence is about 4 to 7 per million per year. Of course, it's mainly a disease of mid to late life. So if you look at younger people under the age of 65, that incidence is under 1 per million per year. However, on Sunday, when we talk about advocacy and going to Capitol Hill on Monday, we're not going to use the 1 in a million number, because what else does 1 in a million mean? It's a rare disease, right? Just by definition, oh, that's so rare, it's 1 in a million. It is a rare disease, but I think it's misleading to say that. So we're going to use what's called the frequency, and that is if you look at the number of deaths that happen every year due to CJD, about 1 in 6,000 deaths are due to CJD. That also equates to a lifetime risk of CJD. So again, these aren't common illnesses, but you certainly should be seeing them. And we heard in our sporadic workshop of two ladies that live a mile away from one another that also unfortunately had to deal with this illness. So I know we have some Maryland people here. The Orioles play about an hour north of here at Oriole Park Camden Yards. It's a small to medium-sized MLB stadium that can house up to 45,000 people. So if we think about a packed stadium, again, we would expect about 7 people in that stadium will develop CJD in their lifetime. So again, not a common disease, but certainly something that you can come across. So that leads to the question, like with our ladies in Southern Maryland, about clusters. Clusters do happen. One of the things about rare disease is if you have 1 case, you have 1 case, but if you have 2 cases, it looks like you have a cluster. So we're just going to briefly talk about how we handle clusters, and you'll hear a little bit about this from Dr. Maddox from CDC tomorrow as well. Clusters are primarily investigated by CDC and local health departments with our assistance as well at the Prion Center. Some steps that we take to investigate these clusters. First, we have to verify that it's prion disease. You'd be surprised, a lot of times clusters reported to us, and a fair number of cases in that cluster wind up actually not even being prion disease. You also want to know the type of prion disease. So for example, families tend to live around one another, and if you have a genetic family and multiple people have gotten sick recently in that family, it's not really a cluster because you have, the known etiology is genetic. You want to verify the numbers being investigated are higher than what's expected. Believe it or not, that 1 to 2 per million per year, you can have quite a variability in it. So you want to make sure that it's not outside of what we would expect. And then you want to verify the likelihood of a common exposure. It's actually quite difficult to transmit prion disease, even in the laboratory sometimes. Basically, you have to have brain tissue from an affected individual that either is injected into another brain of another individual, consumed in large quantities, or injected into someone in large quantities. And that generally does not happen, and not even routine medical care. So we have to make sure that we actually have a viable exposure, not just that someone lived around someone else. They have to have something that makes sense. And then one of the unfortunate things about prion disease, the acquired forms, is that the incubation period can be quite long. So generally it's measured in months to years to decades. So, exposure of about a week or incubation period of a week does not make sense with what we know with the disease. So that's just an example of some of the things that we do with clusters. I do think it's helpful, especially when we hear stories like our Maryland families about clusters. I've had a couple clusters in my life. It's not related at all to my professional life. I live in a fairly middle-sized city of about 30,000 people, and since I've lived there, I've diagnosed about 5 cases of prion disease within about a 5-mile radius of my house. My research nurse was in the son's wedding of one of them that lives down the street from me. My daughter's dentist brother-in-law had CJD, so every time we go to Strides for CJD in Akron, we say hi to my daughter's dentist because she's there. Incidentally, the social worker that saw her brother also went to him for physical therapy when he was well. I went to a friend's funeral, and when I was there, one of his friends came up to me and said, "My grandmother just died of CJD. I have this autopsy report that I can't interpret. Can you help me?" Completely unrelated to my job. I saw an elderly lady for Alzheimer's disease in my memory clinic. Her daughter actually developed prion disease, and that daughter happened to be friends with someone who works at the Prion Center's husband. Former department chairman, his mother died of CJD. And then recently, very bizarrely, a beloved teacher in my daughter's school community died of CJD. And I saw this individual who used to schedule an annual visit to Italy for the Latin class. I literally saw him the day before my daughter went on that trip. So it was quite bizarre. So these are all examples of CJD, 7 degrees of Kevin Bacon, whatever you want to say it. It happens. You're going to see this, right? It's unfortunately a horrible illness. You talk about it, you're going to run across other people that also have had experiences. So how is prion disease diagnosed, and why does it take so long, as we heard in the last session? The diagnostic process really is not unique to medicine. It's not so much different from a mechanic or an IT person. My father was a mechanic for the post office, and he used to always say that us in medicine had it very easy. We only have 2 models, males and females. He had Ford, Chevys, Pontiacs. In a way, he's kind of right. But it's the same diagnostic process, really. Medical diagnoses, just like these other professions, are really typically started by looking at syndromes. So a syndrome is a collection of signs and symptoms that help point to an underlying cause or a problem. And recognizing those syndromes helps the clinician know what to do. So a perfect example is showing up into the ER complaining of chest pain. All right? Chest pain can be due to a lot of things. It could be a heart attack, pneumonia, a pulled muscle. But you have to look at the constellation of symptoms that go along with that chest pain. Is the pain traveling down the arm? Do they have a cough? You also have to look at the context of the history. Were they recently overexerting themselves and it's a pulled muscle? It's the same exact process with prion disease. So if we're going to take the example of, maybe not today, but in another day of trying to understand why your computer is not working, what's the first thing you do? Turn it off, turn it back on. Okay, so you do that, of course that never works. So then you have to go to the next step. Is the monitor plugged in? Is the keyboard, is the mouse plugged in? Is the hard drive broken? Is there something wrong with the software? It's the same process clinically. You just go down the list and try to eliminate all the different variables until you know kind of what's going on. So I'm going to run you through an example of what a patient's initial evaluation might look like with prion disease. So this individual presents to the doctor with some confusion. The memory isn't as good. The doctor asks them a bunch of questions to try to delineate what's going on. They do an examination. And then the doctor's going to be left with a list of possible diagnoses. It could be Alzheimer's disease, vitamin deficiency, thyroid problems, depression. Pretty long list with a lot of different possible etiologies. And then they're going to order a variety of different tests to try to get at that. So they may do some paper and pencil testing of that person's memory, maybe some blood tests, an MRI. And what they're trying to do is they're trying to whinny away at that differential diagnosis, that middle list of conditions. So unfortunately, they do all that. Nothing comes back as positive that help the physician know what's going on. So the patient comes back for the second visit. Well, now, in addition to that person having some confusion, now they're falling a lot. They're complaining that their balance isn't good, that they're walking like they feel like they're drunk. Well, that helps a little bit because now you have a different symptom that can be evaluated. Now you look at things like blood vessel disease in the brain, infections, inflammation of the brain. Go back to doing a variety of other tests. You may repeat some of the ones that were done the first time, but more importantly, you're going to add some that were not included in the first one. They may include different blood tests, a different brain scan, or a spinal tap. Again, you're ruling out potential causes and looking for a result that is suggestive. We do have diagnostic criteria to help us diagnose prion disease when someone is still living. I'll briefly go over that now. There's the old criteria and the new criteria. The old criteria definitely relies on that clinical syndrome. Of course, the only way to definitively diagnose the disease and know what caused it is by autopsy. But for someone who's still living and you're trying to get a clinical diagnosis, again, you look for that clinical syndrome, which has to include cognitive issues, myoclonus, which is that jerky-like movement that some patients get. They can have incoordination, balance problems, or vision symptoms, as we heard about in the last session. They can have weakness or Parkinson's-like symptoms. And then at the very end, they can develop something called akinetic mutism, which is the lack of volitional speech or movement. Not really that helpful for diagnosis because at the very end of the illness. But basically, the clinician is alerted to the possibility of CJD by looking at that list of symptoms and then trying to tie it to a suggestive diagnostic test. And that could be a specific pattern on something called electroencephalogram, EEG, which looks at brain waves. It can be looking at different proteins in the brain, one of them 14-3-3, which we'll talk about the problems with that later. And then the brain MRI, which has really, I think, been a very helpful tool in the arsenal of diagnosing prion disease. But really, the game changed in about 2019 when we started doing a test called RT-QUIC, standing for real-time quaking-induced conversion. In our country, we do that on spinal fluid. This test actually detects the abnormal prion protein in the brain fluid itself. So it's extremely specific for the disease. You wouldn't expect that in anything else. It's so specific that you don't need that clinical syndrome. You just need any neuropsychiatric symptom. There's really very little other neurologic illnesses that are diagnosed like that. And that's because of the specificity of that test. However, ironically, you need to have enough of a suspicion of CJD to know to order that test because you're not going to do that test in everyone. So typical symptoms of human prion disease, we heard about this in the last session. Really, there's no rule for this, right? I mean, it can be tinnitus, it can be imbalance, it could be psychiatric symptoms. There's really a huge litany of symptoms that patients can experience. Cognitive impairment is a very common one, especially early on, but problems with their walking, abnormal movements like tremors or jerky-like movements, visual disturbances. We heard about a lot of that at the last session. Muscle weakness or psychiatric symptoms are extremely common, whether it be seeing things that aren't there, or problems sleeping, mood changes, personality changes. The problem with CJD is that people don't present with that clinical syndrome of CJD when they walk into the office. They have to present usually with one symptom that then evolves into two symptoms and then three or four. If you look at how people present to that initial evaluation, most people will present with kind of a nonspecific cognitive symptom like memory problems. Most people are not going to think of CJD when that's the only symptom. At least they shouldn't, right? Because there are much more common things that they should be thinking of. 17% will present with that pure visual symptom that we heard about earlier. 15% will actually present with some type of mood or anxiety disorder. Only 13% will present with that kind of classic CJD that the doctors will read about in the textbooks with memory problems, problems with balance, and that jerky-like movement. So the thing is, it takes time for the patient to kind of evolve into that clinical syndrome that is going to suggest to the clinician maybe that's what's going on. So we talked a little bit about prion disease tests. We have three big ones in our field. There's the electroencephalogram, which has very specific patterns sometimes for CJD. we have the brain MRI, and then we have the spinal tap. The EEG is nowadays mostly helpful for ruling out other things like seizures. Sometimes it can be suggestive of CJD, but not in all cases and only during certain time points. We really don't use it for diagnosing CJD nowadays. The brain MRI, fortunately, is present in most cases. It definitely narrows down the possibilities of what could be going on. And it may be the first thing that suggests CJD. So if you go back to the individual that had memory problems for the first evaluation, if read appropriately, most of those cases during that diagnostic workup with the MRI, it will be positive. And if it's read correctly, that may be the initial symptom that the patient has CJD, even without all the other symptoms, right? And then that leads the clinician to do the RT quick test to get the confirmation. The spinal tap can help rule out other causes, importantly treatable causes, things like inflammatory conditions, infections, some markers that can suggest CJD, and that one marker, RT quick, that is almost only seen in CJD. Some other terms that you're going to hear about this weekend are references to diagnostic tests. That includes sensitivity, that basically measures how well does a test find the disease that you're looking for, and then specificity, which is how well does the test only find the disease that we're looking for. Many people said, well, I knew that my doctor really suspected that it was prion disease, but they didn't tell me. One of the good things about RT Quick is its specificity. You're not going to find it positive in pretty much anything else. So what we've seen is that really helps the clinician feel comfortable giving the diagnosis with that RT Quick. The double edge to that is that it's not positive in every single case. So now we have clinicians trying to track down that positive RT Quick in cases where there might not ever be a positive RT Quick. But in most cases, that RT Quick has really helped people give the diagnosis much sooner than they would have before. Whereas before, say a decade ago, they would have waited for everything to come back negative before they gave the diagnosis. So just a couple other caveats. The EEG is not very specific or sensitive, but it's fairly specific, meaning that if you see that pattern, it almost certainly is CJD. Luckily, the MRI is very sensitive. It's present in most cases, and it's fairly specific in the right hands. And then depending on what tests you're looking at in the spinal fluid, it's pretty sensitive. Some tests are very specific, and one is extremely specific. That's the RT Quick. Unfortunately, you need some local expertise to read the EEG correctly. The MRI, you have to be able to recognize it. That's getting better, but slowly. And then for the spinal tap, the doctor has to know to order that specific test. In the spinal fluid, we have two markers of neuronal injury, 1433 and total tau. Basically, those are very good at maybe giving you a sense that it could be CJD, but it doesn't mean that it is CJD. And then the RT Quick, which is very specific for the disease itself. I'm going to go over briefly RT Quick because you're going to hear about some of that used in some of the research. The way it works is they take normal prion protein, PRPC, which is your substrate. We add the abnormal sample, which presumably has the disease-associated form of the prion protein to it, with a special dye that will bind to any fibrils that will form. And then it takes advantage of that second slide that I showed you, of that prion paradigm, where you have that kind of exponential growth of the abnormal prion. And if there is the abnormal prion in there, it will bind to the normal prion protein when you put it in this shaker. The dye will bind to it, and you'll have these curves, which is a positive RT Quick. So it's a way of amplifying very, very small amounts of the abnormal prion protein to the point where you can detect it. There are some limitations to diagnosing prion disease, variability in how it presents. Some knowledge of prion disease is required no matter what. The confidence in giving the diagnosis is a huge one, although I think better than what it had been. CJD needs to be the list of possible causes in order order the right test, with the exception of the MRI, which I see a lot more cases being diagnosed because the MRI is positive, not because the clinician thinks it's CJD by symptoms alone. Unfortunately, I don't think our AI guy is here. I made these slides just for him. But he'll be here later. I'll show it to him. So, you know, AI is going to change things, and it's going to change medical practice. So we heard about, you know, it's hard diagnosing CJD. Can AI be helpful with that? If you want to follow the specific prompts, you can just look at the QR code. But I gave them kind of some details of the last patient I saw. So a 79-year-old white woman initially started having changes in her speech, would frequently drop things starting 4 months ago. What do you think the possible list of diagnoses is? Very, very long list, some of which is insane, like Wilson's disease, but it's there. And then I'm not showing this, but they kind of offered a list of tests that I could order. So I did that. None of them were positive, of course, in my patient. So I said the above workup that you suggested was within normal limits, but 2 months later, she developed issues with executive functioning, vision changes, balance problems, affecting her walking and tremors. So revisit that list that you gave me earlier and let me know what else I should do. So that new list was better. Number 6 is CJD. At least it made the list now, so that's good. So I looked at the list of suggested tests. 14 through 3 was on there. So I said all the above diagnostic tests were normal, except the 14-3-3 and total tau proteins and the spinal fluid were elevated. Revisit the possible diagnoses in order of the likelihood. CJD is at the top. So this is helpful, right? Except for the fact that it diagnosed it in the same exact time frame as the clinician did. Because it's following the same method, right? So even AI needs a certain amount of information. In our field, we often say the most helpful tool in diagnosis is time, because you need to have the evolution of the symptoms. Now, there's a couple really concerning things. One is the initial diagnostic workup they suggested would have bankrupted Medicare if we did it for everyone. And they did not mention RT-Quick. Now, I've done this a couple of times, and I keep telling ChatGPT about RT-Quick, but it's not listening. So if everyone does that here, maybe we can get it to listen. And it's, again, diagnosed at the same time point as the human doctors. But I do think there's still a lot of potential for AI, machine learning, definitely in things like MRI. MRI is positive in 95% of cases. We said it depends who looks at it. But if a person doesn't look at it, if a computer looks at it and recognizes the pattern, we might have higher likelihood of detecting cases earlier. Okay, so why is there so much clinical variability in prion disease? I want to go down the rabbit hole a little bit. These are all the different tests that we do at autopsy to diagnose the type of prion disease. We do H&E staining, which looks at the structure of the brain. We put dyes on the brain to look for the abnormal prion protein. We do Western blot to look for the abnormal prion protein and genetic testing. With that information, we get something called molecular subtypes. And we talked a little bit about this. I think someone even diagnosed their loved one's molecular subtype before they even got the autopsy back. So you can see that it actually tells you a lot about the clinical symptoms. But it is helpful to kind of know where that comes from, because all of you are probably a little bit confused about some of the reports. So one of the things that you get with that information is on position 129 of the prion protein, there's something called a polymorphism, which is kind of a normal variation of something that we all have. We all are either MM, MV, or VV. And then on that Western blot, we can have a type 1 or a type 2 prion protein. So what we do is we add the polymorphism in the second column to the prion protein type in the third column, and we get the molecular subtype, MM1, MV1. You're going to hear these ad nauseum this weekend. That's a little molecular subtype. The reason why that's important is because it looks different underneath the microscope, but very importantly for you all, it really describes what you're seeing in your loved one clinically. So MM1 is the most common subtype of sporadic CJD. It makes up about 60% of all cases. This is the classic form of CJD that doctors read about in textbooks. Unfortunately, it's the quick kind, 4 to 6 months, presents typically with memory problems, problems with their walking, jerkiness. But you have longer duration symptoms as well, people that look like an atypical dementia, atypical Alzheimer's disease. That's the MV2. So when we talk about subtypes, this is what we're talking about, and you're going to hear a little bit about this later. So variability in prion disease, other things aside sporadic is genetic prion disease can look very different. There's over 50 mutations that can cause genetic prion disease. Depending on that mutation, it can look very differently clinically. Variant CJD, the kind due to eating meat contaminated with bovine spongiform encephalopathy, looks very different. Part of that may be because it's a different prion protein. Part of it may also be that it presents in younger people, and sometimes prion disease looks different in younger people. Could be a new prion disease. That's what we're worried about at the surveillance center, and we'll talk a little bit more about that on Sunday. Dr. Maddox will talk about the very real concern about chronic wasting disease in deer as well, and the importance of surveillance for that in our country. So I've gone over, so I'm going to stop there and turn it over to Dr. Watts. So Joel will tell you his personal story. He's a very good friend of the foundation. We're very fortunate to have him. He hails from Toronto, Canada. He's a basic science researcher and is excellent at translating some of this basic science stuff. So Joel.
Thank you to Brian for that very kind introduction, and it's very much a privilege and a pleasure to be back here again at this conference. I think that this is probably the most important scientific conference I attend for several reasons. Perhaps the most important one is that not only am I a prion researcher, but also I have been personally affected by prion disease. My father died of sporadic CJD many years ago, and so this disease has sort of been a part of my upbringing and has shaped both my personal and professional life. So my purpose today is to sort of build upon what Dr. Appleby was speaking about in the clinical aspects and give you a sort of a more sort of an overview of the basic science aspects, the fundamental science aspects of prion disease, with the hope that it will facilitate understanding the research grant presentations tomorrow from the CJD Foundation research grant recipients. And so I've entitled my talk Prion Disease 102 because last year I gave Prion Disease 101, so don't worry if you didn't take the prerequisite, you'll be fine. And I've really tried to tailor my talk to the specific research projects that are going to be talked about tomorrow. So Brian didn't really talk about this, but it's important to note that prion diseases not only affect humans. Obviously, we're all here because CJD or other human prion diseases have affected us one way or another, but these are also diseases that are found in animals. And the three major ones are bovine spongiform encephalopathy, or more colloquially known as mad cow disease, chronic wasting disease of deer, moose, and elk, and then scrapie in sheep. And these are important because diseases like chronic wasting disease are really spreading rapidly in certain areas of Canada, the United States, and occur in free-ranging animals, and their relative danger that they pose for humans is not 100% clear yet. The other thing is that because studying prion disease in a laboratory environment is easier with the animal prion diseases, and so we assume that by studying chronic wasting disease or scrapie that we can learn about the fundamental aspects of prions and then be able to apply that to the human prion diseases. So these are the human prion diseases. Dr. Appleby already mentioned them, and so I'm not going to talk about them further. So historically, prion diseases were thought of as the only type of disease that can come about in three completely different ways. You have your sporadic forms of the disease, such as sporadic CJD and VPSPR. You have your genetic versions of the disease, familial CJD, GSS, fatal familial insomnia, and then you have the versions of the disease that are acquired by or transmitted via an infectious route, and these include in humans variant CJD, Kuru, and iatrogenic CJD. Now, there is recent evidence that maybe prion disease is not the only disease that can also occur like this. There are a few cases of Alzheimer's disease that have been identified now which can plausibly be traced back to a transmitted route. And so this is a very active area of research, and it just kind of points to what I'll end off with today is talking about the similarities between prion disease and other neurodegenerative diseases like Alzheimer's disease. So thankfully, sporadic CJD and most of the other human prion diseases are not naturally transmissible from person to person. It means they're not contagious. But there are prion diseases that are. So the animal prion diseases, chronic wasting disease in deer and elk, and scrapie in sheep, can pass from animal to animal by natural means. We're not 100% sure yet what those routes are, but it's clear that that can happen. And another interesting thing about prions is that they're very sticky, which means that they like to adhere to surfaces, such as metal surfaces, and they're very hard to inactivate or destroy. And so the conditions that we would use to destroy other types of infectious agents like bacteria or viruses typically aren't good enough to get rid of prions. You have to use higher temperatures, higher pressures, or harsher chemicals to get rid of them. And this poses a problem. For instance, if you're using surgical instruments and somebody happens to develop CJD later that had been operated on using those instruments, are those surgical instruments now a risk? And there have been a few cases where that happened. And so understanding how we can decontaminate instruments is very important. It's also important for people who work in a lab. If you have a spill of prions, you want to be able to make sure you can clean it up without any residual infectivity present. And so tomorrow you'll hear a presentation from Dr. Jason Bartz, where he's using that ultrasensitive RT-Quick test that Dr. Appleby spoke about to be able to detect how good are certain techniques at decontaminating surfaces once they've been contaminated with prions. All right, so just a quick biology overview, basically high school biology. So you have DNA. So DNA are our genes. I'm sure most people have heard of our genes. So these are our chromosomes that are found in cells. They're the genetic material. So what I have up on the screen for you right now is what's called the central dogma of molecular biology. So you have DNA, it gets made into RNA, and then RNA gets made into proteins. And proteins are the workhorses of our cells. They can be enzymes that catalyze chemical reactions. They can have a structural role in the cell. They can give the cell its shape. They can help regulate what gets in and out of a cell. They can help cells talk to each other. They can provide scaffolds for other proteins to come together and work together. And so here is the sequence of the prion gene. The prion gene is called PRNP for prion protein. And so it's a combination of the letters A, T, G, and C. It doesn't really mean much when you just look at it, but that gets translated into a protein sequence. Now, a protein is basically a linear sequence of building blocks called amino acids. You can think of them as a string with 20 different colored possible beads, and the arrangement of those beads are what define how the protein folds up into its three-dimensional structure. And the three-dimensional structure of a protein is what governs what it does in a cell, its function. So this is the three-dimensional structure of the prion protein. Those corkscrew-looking things are called alpha helices, and so this is what the prion protein looks like. All right, so in a sort of a reductionist approach, this is prion disease in a nutshell, or what I would call the prion principle. You have two forms of the prion protein. You have PRP-C, or cellular PRP. This is the normal form of the protein. It's present in all of our brains at fairly high levels. And during disease, instead of taking its normal shape, it gets converted into a different shape. That shape is called PRP-SC. SC stands for scrapie, which was the first widely studied prion disease. And so PRP-scrapie is also called just prions. And so PRP-SC clumps together and forms aggregates. The proteins sort of self-assemble into these large deposits that cause dysfunction in our brain and ultimately arise in the death of cells and other dysfunction that causes the clinical manifestation of prion disease. Now, the thing that's unique about prions is that PRP-SC, as Dr. Appleby said, is like the bad kid on the schoolyard. It promotes the misfolding of other PRPC molecules so that you have a net increase in PRP-SC. So this is why prions are infectious. So if you take a pre-existing source of PRP-SC and you introduce it into a new host, it's going to start converting the host PRPC into more PRP-SC, starting off the disease process. So one often wonders, why do we have the prion protein to begin with? What is the function of PRPC? It must do something useful. And the way you would typically go about trying to find out what is the function of a protein is you get rid of it and you see what goes wrong. You can get rid of the prion protein in mice and cows and goats and maybe even in humans, and nothing really appears to go wrong at the surface. Now, PRPC does appear to play a minor role in many different biological processes, some of which are shown on the screen here. But it's still not 100% clear why we have this protein, what its true role is. Sometimes unexpected things crop up. Like, for instance, there have been recent papers suggesting that prions might also be important, or prion protein, PRPC, may be important for protecting us against certain types of viruses. Now, the reason why we want to know what the function of PRPC is because one of the best ways to prevent prion disease is to get rid of PRPC. If you have no PRPC, you can't make prions. And so is getting rid of PRPC safe? And so tomorrow you will hear a talk from Dr. Victoria Lawson, who is examining sort of the unexpected finding that when you lower or get rid of PRPC, that you might actually increase risk for certain types of cancers. So this is very interesting research that unfortunately I will have to miss tomorrow, but I'm sure it's going to be a highlight of the conference. Okay, so what exactly is a prion? For those of you who were here last year, you'll probably remember this slide. So you can think of PRPC, the normal prion protein, as a Chevrolet Corvette. So prions are like all proteins. They are not actually static. They're dynamic entities. Their structures can fluctuate slightly. So you can change some subtle things to make the protein appear differently. So you can open the car door, you can open the hood, you can put the roof up or down. But essentially, you can still recognize that that's a car. It's the same basic structure. So when I talk about obtaining a different shape of a protein during prion disease, it's conversion to a very, very different shape. So to use this transformer analogy, you get conversion of something that's somehow good in terms of a Chevrolet Corvette until something that's very bad as the evil Decepticon transformer. And so here's the slightly more scientific version of that slide. Again, this is the prion principle, where you have your normal PRPC, shown here, which is mostly made up of these corkscrew structures, which are called alpha helices, shown in red, changing its shape into something that is very different. Now, until a couple years ago, we didn't know what the structure of PRPSC looked like. Now we do. So it's this protein that's composed mostly of these purple things. These purple things are called beta sheets. So in PRPC, there's very little of that. In PRPSC, it's entirely beta sheet almost. And these molecules stack together to form these large aggregates that going to come out at you at the screen here to form these large aggregates. And these are the aggregates that are caused the problems in the cells. Now, just to kind of go over that a little bit in a bit more detail, so just simplify it here, you have PRPC as a blue circle, PRPSC as a red square. Now, to start off prion disease, you have to have the initial production of PRPSC. In the infectious or acquired versions, you don't have to have this. You have a pre-existing source of PRPSC from somewhere else. So we really know surprisingly little about what kind of causes that initial misfolding of PRPC into PRPSC. It could be spontaneous, which means just occurs randomly. We know that mutations in the prion protein in the case of the genetic prion diseases somehow predispose this to happen, but it happens at some low frequency. And usually I think cells can probably deal with it, which is why prion diseases occur more commonly in people who are older, when quality control mechanisms in our brains potentially aren't working as well. So once PRPSC is formed, when it encounters a molecule of PRPC and it interacts with it, it causes production of a second copy of PRPSC. So now you've got two molecules of PRPSC, and you can think of this as like a Lego block where you stacked two 2x2 Lego blocks on top of each other. So then if you want to start building upon that, now you've got two ends. You can either add to the left side or the right side, the top or the bottom. So you add two more PRPC molecules, they get converted, and so on and so on. And this is how prion aggregates start to grow. And then eventually these are going to get bigger and bigger. They'll get broken up into smaller pieces, which allows for these sort of exponential amplification. And these smaller aggregates, which are probably the type that occur early on in the disease course and are actually thought to be maybe the most infectious and the most toxic, or what are called PRP-scrapy oligomers. So several copies of the prion protein stacked together. These can then grow into be bigger aggregates, which are typically called fibrils. And so tomorrow you will hear a presentation from the multi-talented Dr. Valerie Sim, who will talk about her research specifically looking at these oligomeric types of PRPSC, so the smaller aggregates and how they relate to disease in VPSPR and other types of genetic, or in other genetic, or in genetic prion diseases. So, Dr. Appleby mentioned that PRPC is sort of like the only player in prion disease, and for all we know, currently that's true. But there's always been a question, could there be other proteins involved? Could there be proteins that either help promote the initial production of PRPSC or to prevent it from happening? Or could there be proteins that help with prion replication? And the reason we would really like to know this is because if there were such proteins, that would be something else we could think about targeting with a therapeutic to help prevent or delay prion disease. And so two of the talks you're going to hear tomorrow are from researchers who are taking very different approaches, but to tackle the same question. Basically, are there other proteins that are involved in prion disease? And so first you'll hear from Dr. Robert Mercer, who is going to talk about trying to find proteins that bind specifically to PRPSC. We know many proteins that bind specifically to PRPC, and none of them so far have been involved in prion replication. However, by identifying molecules that bind to PRPSC, he hopes to apply the guilt by association principle, and that if a protein binds to PRPSC, that it's probably most likely that it somehow is related to prion replication. The other is going to be from Dr. Thomas Cunningham, who's using a totally different approach. So based on some of the beautiful genetic work done by Simon Mead, I'm sure you'll hear a little bit about this in his keynote tomorrow morning, is that due to genetic variability amongst the human population, he has found certain variants in other genes that are more commonly associated with people who have sporadic CJD than would be inferred to happen by chance alone. And one of these genes is called STX6. And I mentioned earlier, one way you want to tell what happens to a protein's function is you get rid of it and see what goes wrong. The other way is you make more of it and see what happens. And so Dr. Cunningham is going to talk about prion pathogenesis in mice that express more than the normal amount of STX6. These are so-called overexpressing transgenic mice. Now, I told you a few minutes ago that PRPC is a blue circle and PRPSC is a red square, and it's not actually that simple. So there are actually multiple types of PRPSC. So PRPC can misfold in several different ways to give different types of PRPSC, and these are called prion strains. So I'm sure many of you have heard of the concept of strains before in the context of other diseases. So for instance, with viruses, you can have different strains of viruses like the influenza virus, like COVID, that because of changes require you to get a new vaccine every year to kind of keep up with the currently circulating pool of viruses. Same sort of thing with prions, except instead of changes at the DNA or the RNA level, this is changes at the structural level. So different structures of PRPSC give rise to different prion strains that then cause different phenotypes in animals or humans. Now, in a research lab, how would we distinguish between PRPC and PRPSC? And you're going to see a lot of this in the talks tomorrow, I'd imagine. And so the most common way of doing it is to use a protein or an enzyme called proteinase K. So a protease or proteinase is an enzyme whose job it is to chew up other proteins so that the amino acid building blocks can be recycled for other purposes in the cell. So proteinase K, or PK, when it encounters PRPC, it can chew it up without issue because PRPC is a relatively small protein. You can think of it, it can fit inside Pac-Man's mouth. However, when Pac-Man PK encounters PRPSC, because these are aggregates of proteins where you have many copies of the same protein stacked upon each other to create one of these big rods, it doesn't fit in the enzyme's mouth anymore. And therefore, it can't replicate or it cannot chew this up. And so we use this enzyme to distinguish biochemically between PRPC and PRPSC. And so you will see lots of things that are called Western blots or immunoblots where you separate proteins on the basis of their size, and then you use an antibody to specifically go and find the prion protein. And after digestion with proteinase K, you will see all these different patterns that you see up there on the screen. And each of these different patterns is a kind of a signature for a different type of prion disease. So I've shown here what the prion protein looks like after PK digestion in 4 different types of human prion diseases. And the basis of this is because each of these diseases is caused by a distinct structure of PRPSC. And this is really amazing work that has come out in the last few years from our colleagues in London and at the Rocky Mountain Labs as well, using cryo-electron microscopy to solve the structure of prions from brains. So here are 2 examples of 2 different strains of mouse prions called ME7 and RML. So at first glance, you would look at these and say, Joel, these are identical. They look the same. And they are very similar, but there are sort of subtle differences. Like if you look at the parts that are purple and green in the 2 pictures, those parts of the protein are slightly different from the 2. And these are enough to cause very different changes into how the prion behaves in mice. What we don't know yet are the structure of prions from humans, especially CJD cases. And why does this matter? Well, there are some fundamental differences as far as we can tell between mouse prions and human prions. And so it's probably likely that if we're going to be able to say, use these structures to design drugs that combat or eliminate prions, we need to know the structure of actual human prions. And so that's the goal of Dr. Chuye Lee's presentation tomorrow. Dr. Lee is the inaugural recipient of the CJD Foundation Postdoctoral Research Award. So he's going to talk about his efforts to use cryo-electron microscopy towards elucidating the structure of different types of human prions. Now, the most common way we study prions in a laboratory environment is to take advantage of the fact that prions are infectious. And so we can take a pre-existing source of prions and we can inject them into laboratory rodents, such as mice or hamsters. And then we're going to wait for a while, and eventually those mice or hamsters are going to develop prion disease. So depending on the species, will depend how long it takes. So mice, only it takes about 4 to 5 months after you inject them with prions for them to develop disease. Hamsters, much faster, less than 3 months. And you say, why don't you use hamsters all the time? Well, hamsters are a bit of a pain to deal with in the lab, and they're more expensive. And mice you can genetically manipulate much more easier than hamsters. And so mice have really been the preferred laboratory animal for studying prion disease. And if you take the brains of mice, that either healthy mice or prion-infected mice, and you apply this proteinase K digestion assay that I talked about. So in healthy mice, this black signal you see on the left here is PRPC. When you treat healthy mouse brains with proteinase K, all the PRPC goes away. When you treat prion-infected mice with proteinase K, a portion of the protein remains, and that's PRPSC. And if you look at the brains of these mice, they look identical to what you would see in a human prion disease case. So they have the spongiform chains or vacuolation, the holes in the brain to give it this Swiss cheese appearance. Exactly the same you see in animal and other animal and human prion disease cases. These brown clusters or dots, these are deposits of the abnormal prion protein, PRPSC. And so as prion researchers, we are very lucky that we have incredible animal paradigms for studying prion disease. Researchers who study other brain diseases like Alzheimer's disease and Parkinson's disease, they're not so lucky. Now, I've talked about prions being infectious, but this is not uniformly the case. So mice are very susceptible to prions from other mice, but they're not so susceptible to prions from other species. So let's say if I took mice and I injected them with 20 mice with prions from mice. All 20 mice are going to get sick, and it's going to take 4 to 5 months for them to do so. However, if I inject them with prions from another organisms, like hamster prions, human prions, etc., then you're going to get very poor transmission, meaning that only maybe 1 out of 20 mice gets disease, and instead of taking 4 to 5 months, maybe it takes 2 years to do so. This is because of the species barrier, which basically means that prions from one species are not so good at infecting a different species. Now, mice and hamsters, the species that are used in a lab commonly, are somewhere in the middle of the spectrum. You have at one end animal species that are quite resistant to prion disease, such as horses and dogs. This is because of only a few changes in the amino acid sequence of the prion protein causes you to make a prion protein that doesn't like to form PRPSC. Whereas at the other end of the spectrum, you have the bankful. Bankfuls are susceptible to prions from every species without much of a species barrier effect. All because, again, they have a researcher, a lucky or a bankful, unlucky combination of specific amino acids that make it very common to adopting the misfolded version. And so I've always thought, because I work on bankfuls, that the bankful is really the superhero of the prion disease field. Now, I just wanted to have one slide about RT-QuIC. Dr. Appleby already introduced this and explained how it's really revolutionized diagnosis in the human prion diseases. Here's how RT-QuIC works. And so you take recombinant PRP, which is produced in bacteria, and then you add to it a source of prions. And then you put it in one of these plate readers, and you add the dithioflavin T, shown down here in the bottom left. It's basically just a dye that becomes fluorescent when it becomes bound to protein aggregates. And you shake these samples, and eventually, in red here, if you've used a sample from a prion-infected mouse, then the signals start to go up, and you have a positive test, where if you use a sample from a healthy mouse brain, then the signal stays flat. The important things to note about RT-QuIC, the animal bioassays I told you took like anywhere 3 to 5 months. This can give you a result in like 5 to 10 hours. So it has really, really improved the efficiency of prion disease research. The other thing that's fantastic is that it's exquisitely sensitive. Dr. Appleby talked about using it to detect prions in CSF, cerebral spinal fluid from patients. You can also do it from nasal brushings, from skin biopsies, even from CJD patients. However, these are still somewhat invasive samples to obtain, and so we're always looking for new ways to obtain samples that are less invasive. And so Dr. Sarah Vassalari, which you'll hear about tomorrow, is trying to develop blood and urine tests for prion disease using this RT-Quick system. Most of us here are obviously very interested in the development of prion disease therapeutics. So going back to this prion principle, there are several parts where you could potentially, or points where you could potentially intervene. You could get rid of PRPC, because if you have no PRPC, then you can't make prions. And this really is the basis for several ongoing efforts in the field, including the clinical trial that's being run by Ionis. You could inhibit conversion of the prions. You could get rid of PRPC once you've made it. You can block it from being toxic, or perhaps some combination of all four of these. Now, mice are really good if you have a therapeutic in mind at testing whether or not that therapeutic works. The problem is mice are not good if you want to try and screen hundreds or thousands of potential therapeutics to see if they work. For that, you need a simpler system. And that's where replicating prions in cells comes in. So just like mice, you can take cells that are in a petri dish, you can expose them to prions, you can passage the cells several times, and eventually they're going to become infected with prions, as evidenced by the presence of proteinase K resistant PRP in your cells. And this is really, this ability to propagate prions in cells has really facilitated our understanding of the biology of PRPSC and allowed us to identify drugs that are good at inhibiting mouse prions. Unfortunately, replicating prions from other species, including humans, in cultured cells has proven to be much, much more difficult than we had hoped. And so this is one of the sort of major limitations, I think, that is hindering us from having a therapeutic for prion disease. And that's why I am very, very excited to hear the research tomorrow from Dr. Paramjit Jat from London, who is talking about his efforts, or who will talk about his efforts to propagate human prions, variant CJD and sporadic CJD, in cultured cells, and how hopefully that those will provide a paradigm for identifying therapeutics that are specifically active against CJD. All right, so just to conclude my talk, I want to put prion disease in a perspective compared to other brain diseases. And so I'm sure, as most of you know, there are other neurodegenerative diseases, such as Alzheimer's disease and Parkinson's disease, ALS or Lou Gehrig's disease, frontotemporal dementias, Huntington's disease, all of which are quite a bit more common than prion diseases. And there are a lot of similarities between these diseases, perhaps more than you would think. So on the surface, they're all neurodegenerative diseases, so they all involve loss of brain cells and create problems with your central or peripheral nervous systems. They all involve protein misfolding and aggregation. And perhaps interestingly, and something that's becoming more recognized in recent years, is because most of these diseases affect or are more common in people as they get older. It turns out that people who have Alzheimer's disease pathology in their brains, actually a good proportion of them also have, say, Parkinson's disease pathology in their brain, and the same vice versa. So people who develop Parkinson's disease also have signs of early Alzheimer's disease frequently. So what about for prion disease? Well, prion disease, yes, you also, because it occurs mostly in people who are older, you also have evidence for other types of pathologies in the brain, such as that which would be indicative of Alzheimer's disease. But what about the opposite? Do you ever get in a non-prion disease evidence for co-pathology of PRPSC? And this is what Dr. Ignazio Calli is going to be talking about, looking for whether in other dementia diseases such as frontotemporal dementia or Alzheimer's disease, do you see any evidence for the production of abnormal forms of the prion protein? Again, using this ultra-sensitive RT-Quick test. So lastly, just returning once again to the prion principle, it is now recognized that this can be generalized to apply to most, if not all, neurodegenerative diseases. So instead of PRPC going to PRPSC, you have some normal protein or peptide being converted into a misfolded version that then self-assembles, aggregates, deposits in the brain, and causes problems. And it's now been shown that just like prions, Proteins like A-beta in Alzheimer's disease, tau in Alzheimer's disease, alpha-synuclein in Parkinson's disease. These proteins can exhibit quote-unquote prion-like properties by being able to stimulate the production of more forms of the misfolded version of the protein from the normal version. And this has really allowed us to enhance our ability to understand and diagnose other neurodegenerative diseases. So the RT-QuIC test we talked about for prion disease, it has now been developed versions of the RT-QuIC test that can use to develop or diagnose Alzheimer's disease or Parkinson's disease in patients while they're still alive. We can now have animal models for Alzheimer's disease and Parkinson's disease where, like for the prion models, where you inject them with a seed of protein aggregates that then causes protein aggregation in an animal model that you can use to study how these aggregates spread in the brain. And the idea that strains of protein aggregates explain protein or clinical and pathological variability in neurodegenerative diseases has now shown to be true not only for the prion diseases, but also for diseases caused by alpha-synuclein and diseases caused by tau. And so I'll end with by saying that fundamental prion research, because all of this has been rooted in our understanding of prion diseases, has had a disproportionately positive effect on not just our ability to understand, diagnose, and treat prion diseases, but to our ability to understand, diagnose, and treat other more common neurodegenerative diseases as well. And so with that, I'll end, and then Brian and I will happy to take any questions. Thank you.
So those of you who were here last year will be aware of this, but the CJD Foundation has kind of been on a crusade to have Alzheimer's disease and related dementias include prion disease in their club of funding. And in part, that will happen in November. We're going to have a huge conference here in this area in Bethesda, invite NIH to try to convince some of the key funding leaders of the field to include us, which will help us, of course, but I think you make a good point, Joel. We really are helping these other neurodegenerative illnesses. So I'm sure, Debbie, you'll talk about that later as well, but I just wanted to make sure that I accentuated that point since you ended on it.
Got a question?
Oh, yeah, thanks. Yes, I was wondering, does a history of Alzheimer's disease make a person more susceptible to CJD?
Not that we're aware of. There is some evidence that a very specific form of prion disease called variably protease-sensitive prionopathy, sorry, we don't make these words up, they may have a slightly increased risk of other progressive brain illnesses, but it doesn't seem to be a very key or strong risk factor for prion disease in general.
And I can tell you that in mice, people have tried to ask that question by using mouse models of Alzheimer's disease and seeing if, for instance, does prion disease happen faster in these mice, and it doesn't. So probably there's not a lot of interaction, generally speaking, between the two diseases.
When you talked about the different subtypes, and in the sporadic CJD meeting, it came out that there was some individuals that had multiple subtypes, or, in our case, Natalie had like MV1-ish, right? Like not really super definitive of one subtype. Like, is that a common thing, or will there be like additional subtypes to kind of narrow that down and maybe, zone in on maybe differences?
Yeah, so when we were talking about in the sporadic workshop, multiple subtypes, what we're generally referring to, if you remember how the molecular subtype formula is created, it's the polymorphism, MVVV or MM, plus a type 1 or type 2 prion protein. But 30% or more of all prion disease will have more than one prion protein type, so type 1 or type 2. So when people were talking about multiple types, they were talking about, say, for example, MV1 plus 2. So that happens in about 30% of cases. Natalie, unfortunately, is unique in many respects in that that's not necessarily an example of multiple subtypes, but just a rarer subtype than what we typically see. So as you know, there's only really a couple other cases that were described that kind of resembled what Natalie exhibited. So.
Can you tell by the subtype, and maybe I'm misunderstanding what a subtype is, how a person is affected by CJD or how long it will last?
You can. So subtype is generally only found out at autopsy, but we do have some ways of guessing at the subtype while someone's still living, and that includes things like how they presented clinically, symptom-wise, what their course has been like clinically, and also what their brain MRI looks like. But the only way to definitively know what subtype it is at autopsy. Some of that information on a lumbar puncture may be useful at coming to a subtype, but it's not as helpful as those other variables I talked about.
I heard once that maybe a stressful or traumatic event could trigger the prion disease to show up. Is that true?
That's a tough question to answer. I don't know if it's answerable. I can tell you kind of three different scenarios in which might explain that. Number one is the number one risk factor for sporadic prion disease is age. Unfortunately, people's health decline as you get older, so they're more likely to have things like medical procedures or medical illnesses around the time of their presentation of CJD. The second issue is the chicken and egg effect. So a lot of times people will say their loved one had a lot of stress at work, they weren't doing well, they were getting really stressed out, or they fell. It's hard to say whether or not that was a precipitating factor or the initial symptoms of what was going on. Were they more stressed out because they were having difficulty coping, because they were having subtle cognitive issues? And then, other possibilities, include, we just don't know. But plenty of people have stressful life events, other medical conditions. If it does increase someone's risk, it's, not the one thing that does it. I don't know. Is there a mouse model for that? Has anyone looked at hitting mice over the head beforehand or anything like that?
I mean, there certainly are mouse models of that, because there are other neurodegenerative diseases like chronic traumatic encephalopathy that are thought to be caused by repetitive instances of mild traumatic brain injury, concussions basically. And so there are animal models where you can recreate that. I do not think that anybody has ever done that and looked what happens with prion disease, to the best of my knowledge.
Actually, Ignacio Cali here did a very interesting study that looked at chronic traumatic encephalopathy, the brain disease of football players and hockey players. And he found that in prion disease patients, there was a greater than expected amount of people that also had that. We're still talking about very, very small numbers, but that would suggest that maybe things like head injury may somewhat predispose people to prion disease. Now, I think it's safe to say head injuries are not good. So, plenty of people have head injuries and don't develop prion disease or these other progressive brain diseases, but it may be one variable that helps pushes someone towards that.
Brian, I was just going to add that we looked at this in UK. We did what's called a case control study. We selected all the people with prion disease, and we looked back at things like divorce, losing your job, a diagnosis of anxiety, head injury, all of these sort of things. And then we looked at people who had other neurological diseases like multiple sclerosis or whatever. And then we looked at other people who'd been admitted to hospital for routine surgery like a hernia repair and so on. And we found no difference in incidence of these events between the three groups.
Hi. Can you explain what's meant by proteins folding or misfolding?
Sure. So a protein, when it normally gets synthesized in a cell, you can think of it as a linear chain. And these linear chains don't really in most cases, don't really impart any sort of function to the protein. So it needs to be able to become more compact and obtain a three-dimensional structure. And this is called protein folding. And this is a thermodynamic process that is governed by things like entropy or enthalpy and things like that that you will remember from university chemistry if you ever took that. And so a protein, typically what has long been thought is that a given amino acid sequence, so a given protein, only folds into one structure. Prions are an exception to that rule because they can fold into at least two different stable structures, the PRPC and the PRPSC. So misfolding refers to either starting with the normal protein and somehow going through a refolding event so that it becomes a structure that is not the normal one, or right from the start it kind of folds into the non-native structure. So that's what folding and misfolding are.
Hi, I have a question. So we all have, as I understand, we all have the PRPC protein, and we create it naturally. Do we know how does the body, or if the body has a pathway or a mechanism to degrade it naturally? Like, do we always create this protein? And if we do know how the body is degrading the protein naturally, can we learn from that mechanism and kind of use it to degrade the protein when it's like being misfolded?
Yeah, that's a fantastic question. So yes, so you don't make prion protein at birth and then have the same prion proteins for life. No, it's continually made, synthesized in cells and turned over. So using cells or mice, one can estimate how long a single copy of the prion protein would live. And it's somewhere in the 16 hours to 1 day range. And so every couple of days, you're kind of producing a new set of PRPC, which means that the pre-existing set, yes, does get turned over. And that's through various pathways in the cell, such as the endosomal lysosomal pathway. And so theoretically, if you wanted to pursue the strategy of preventing prion disease by reducing the levels of the PRPC substrate, then you could try and stimulate those pathways so that you end up with less steady state levels of PRPC in the brain. And that has been tried before, although I'm not sure that it has progressed to the level where it could be tested in humans. Now, in terms of degrading PRPSC, cells can also get rid of PRPSC. Of course, it occurs much more slowly than getting rid of PRPC. But for instance, if you take mice where you infect them with prions and then they've been genetically engineered in a way so that you can then shut off PRPC so it's no longer made, those mice will get rid of the PRPSC that they've already accumulated. So you can get rid of it, and actually not as much is known about the pathways that get rid of PRPSC. But yes, theoretically those could be tapped into, at least as a complementary strategy for prion disease therapies. Somewhat. So immunotherapy is the question. Is the so the CRISPR the CRISPR is something very famous in the news these days. So CRISPR is a gene editing approach, and so CRISPR was identified in bacteria as sort of a bacterial immune system to prevent itself from being infected with viruses repeatedly. And people have modified this to be able to edit genes in cells. And so CRISPR has the potential of, say, going in, and if you have a mutation in your prion gene that predisposes to genetic prion disease, you could theoretically go in and potentially correct that mutation. Or you could use the CRISPR to go in, prevent the prion protein from ever being made itself. So this sounds fantastic in principle, but there are some very big logistical barriers. One is that getting the machinery, the CRISPR machinery into the brain is hard to do because it has to go through that pesky blood-brain barrier. The other problem is that the machinery is quite big and they don't fit very well in some of the viruses that you would use in gene therapy. The other problem is that prion replication probably is most important in neurons. So that's our kind of this type of cell in the brain that is responsible for communication with other cells. And neurons do not divide. They're not, once a neuron is made, it doesn't reproduce to make more neurons. And so those types of cells are harder to do gene editing in, and you can't repair it using the technology as well. And so there are, I think, hopefully that we will eventually get there, but there are still some challenges that need to be overcome for gene therapy for prion disease.
To your question, to your point, the person sitting behind you, Simon Mead, did an interesting study. He can correct me if I'm wrong in how I say this, but he looked at different genes in sporadic CJD outside of the prion protein gene to see if there are any differences or similarities between all the cases. And he found that there was a specific gene, STX6, that there seemed to be a common variant shared not only amongst sporadic CJD, but another progressive brain disease called progressive supranuclear palsy. And that gene also is implicated in one of those clearance mechanism pathways, ubiquitin process. So it could be that there are issues clearing these proteins that they may have similarities, not just in prion disease, but other neurodegenerative illnesses as well. Will you be talking about that tomorrow morning at all?
Thanks, Brian. Tom Cunningham will be talking about that much more than me. I will refer to it in a few slides, but Tom's going to cover STX6 in detail, yeah.
Hi guys, fantastic primer. When you read a research paper or you listen to a scientific lecture, how do you decide if you believe the results the researcher is sharing, or if you're a little skeptical, or if it's just too early and we don't know yet?
I think this gets easier with experience, because as the more experienced you become, you've had a more chance to do research yourself. You've had a chance to see stories that passed the test of time and those that didn't, and I think that helps evaluate things that look really promising or stuff that is just smoke and mirrors or hot air, basically. It's not easy. To me, a key thing is if you have somebody publishes a really exciting but perhaps controversial study, that seeing it replicated by an independent group is when I really then start to believe in it.
Maybe I'm a natural skeptic, but I like to see when people really try to disprove what they found, right? A lot of research sometimes will just try to prove a point, but when you actively try to disprove that point, I think that's an example of a very good study.
Is there a specific part of the brain where CJD might start commonly, or do we know about anything like that? Is there any particular, could it be anywhere in the brain? Start anywhere and go anywhere, and what goes on?
Yeah, it can start in a variety of different areas of the brain. That's partly where these molecular subtypes come in and why people have different symptoms during different parts of their illness. In neurology, clinical symptoms is kind of like real estate. It's location, location, location. Depending on where the pathology is where you're going to see the symptoms. Going back to AI for a second, Dr. Beasy, who is the gentleman who reads all that brain MRIs for the brain MRI consultation program, was able to use all those MRIs to kind of look back and trace back the origin of where a lot of these subtypes start. And he used that using machine learning, AI essentially. But yeah, there's probably about 3 or 4 different origins of where prion disease will start in the brain, if not more.
One of the challenges is that, of course, you don't know who's going to be a sporadic CJD patient until they already basically they're at disease endpoint. And so it's harder to have earlier samples from those individuals. And so ideally you would love to have an animal model to be able to study these questions, and studying the initial or spontaneous formation of prions in mice has been difficult, and it's an area of research that I'm actively involved in, and we're still not there yet in terms of being able to recapitulate all of the earliest events in prion disease and getting mice to kind of go to that similar endpoint that, that you would see in human cases. It has been very difficult, and so it's a really amazing question that I think is going to be a really interesting one to answer, but it's hard to make definitive conclusions at this point.
The way that you talked about like the time that it takes from injecting the prions into the mice and how long it takes for them to get sick, do you have ideas about like how long it takes from a prion misfolding to how long it is until a person shows symptoms? Like, is it years or is it weeks, or, you know, I would assume it'd be longer than the mice and the hamsters.
Yeah, so some of the rarer sort of prion diseases have given us some clues in that. So one instance is Kuru, which is the prion disease that occurred in the Foray tribe in Papua New Guinea that was spread through cannibalism. And so this is an infectious prion disease, and one can reasonably approximate when that process stopped in the population, and then you can look at how long it took the last people to develop Kuru. And based on those studies for these infectious prion diseases, and Simon could correct me if I'm wrong, but it can be upwards of 50 years almost between when you were initially exposed to prions and developing the disease. For the genetic prion diseases, here again you have a population of people who will likely develop prion disease during their lifespan. And people are now trying to use RT-QuIC to predict when those people sort of convert into the prion disease state. And Eric and Sonia, who are in the audience, have been doing some studies, as have Simon and colleagues. And it looks like maybe sort of 3 or 4 years potentially before you can start to detect this sort of abnormal prion protein. But who knows what the earlier events even than that are. And so it could be, you know, decades before. We don't really know.
I mean, if we borrow a page from Alzheimer's disease, we know it essentially is a chronic illness. You see pathology in the brain of Alzheimer's for at least a decade or more before you see the symptoms. So essentially when you see the symptoms, that's the tip of the iceberg. So presumably something like that happens as well with prion disease. It's just how long is that silent stage where you have pathology but no symptoms?
Hey, in the curve from diagnosis to death and the kind of the 1 year steep drop, would earlier testing of RT-QuIC have a relationship to potentially flatten that out? I guess in other words, if doctors are slower and wait for more and more symptoms before they begin thinking about this, is that in any way related to why the curve is so steep at a year?
So that curve was time from symptom onset.
Oh, not, okay, right.
Yeah, so if you were to look at time from diagnosis to someone passing, that would be even steeper. And it's usually about a month on average, with a lot of variability. So.
There's a lot of great research. What's your optimism of finding a cure and, you know, maybe a timeline or?
Yeah, I mean, I'm optimistic that we will have something for prion disease. I think because of the last question, I think once someone's sick, that's going to be difficult, right? Because there's always going to be a delay between the person developing symptoms to the time you get a diagnosis to the time that you start developing symptoms. And we may develop some type of treatment that affects the disease process and halts it, but we don't have anything that can reverse brain cell damage for any disease yet. So it becomes a twofold problem, stopping the disease and reversing the damage. However, it's a very different situation when you talk about people that know they carry a mutation and they're at risk of developing the disease. Because then if you can stop the disease process or delay it, then you don't have to worry about reversing brain cell damage because it hasn't started yet. So with a lot of work of people in this room, I think it's very likely we'll develop something. I don't know how effective it will be, but something within our lifetime that will affect those individuals, hopefully being able to delay or prevent the onset. But I think symptomatic illness is going to be really, really difficult to do something about. You want to comment on that?
I mean, Brian makes an excellent point regarding the reversing the degeneration that's already occurred, and that's going to require advances in regenerative medicine, stem cell therapy, et cetera. I'm not sure. I am really optimistic about the PRPC lowering approaches, whether it be the antisense oligonucleotides, whether it be zinc finger repressors, whether it be CHARM, this really cool new technique that's been developed at MIT. I think that is going to have some effect if it can be given early enough. And so I am optimistic that maybe not in the first iteration of the Ionis clinical trial, but maybe subsequent iterations, that we are going to start to see some preliminary evidence of benefit, similar to what has been achieved in the Alzheimer's field, where in the past years we've had some preliminary evidence that we can create disease-modifying therapies. Now they're not very good yet, but it's a first step. And so I'm hopeful that these first clinical trials, that we're going to get there quicker than the Alzheimer's disease field did. So I am optimistic about that. I think what everybody would love to be able to be a pill, basically, you could take to treat prion disease, and that's usually some kind of small molecule drug. And I think we're a little bit lagging behind in that because we don't have a good way of identifying drugs that work against human prions. And so hopefully when Dr. Jot talks tomorrow, he'll tell us about the advances he made in that area of research, and hopefully that is something that will sort of advance in parallel with the antisense and other strategies. And I'm certainly more optimistic now than I was, say, 5-10 years ago. And so I can't give a timeline, obviously, but I think there's a lot of reason for optimism.
And I think we've reached the end of this session. So the next session at 4 p.m. is bereavement. That will be in this room. And then after that, we'll have the reception. So thank you.